Okay. So far as we've been studying Mendelian genetics, we've been looking at the crosses that Mendel did where he was only following one particular character, right? So one single character, he's just looking at flower color. And he was able then to use his crosses for this one character to be able to come up with the law of segregation. And that we've already gone through, but it's restated here, right? The law of segregation says that two alleles for a heritable character are going to separate or segregate during gamete formation and end up in different gametes. Now, when he was coming up with his law of segregation to explain that 3-to-1 ratio in the F2 offspring. And again, he was studying just one single character at a time. So really, for that series of crosses, the F1 offspring that were produced by the first crossing the parental generation to each other to create those F1 hybrids, those are really called monohybrids because they are heterozygotes for just one character. Mono indicating one, hybrid indicating that they are heterozygotes for whatever character it is that he was looking at. When he took those F1 hybrids and he crossed them to themselves, this is called a monohybrid cross because you're taking the heterozygotes for that one character and you are crossing them to themselves. Mendel didn't stop there. It's like, no, that's not good enough. Well, what happens if I cross hybrids that are that have different alleles for two characters at the same time? And so Mendel was able to identify his second law of inheritance by following two characters at the same time as opposed to just one. So he's making it doubly as complicated. So let's take a look at his double crosses. When he did the double crosses, same concept applies, same series of crosses as he did studying the monohybrids. But in this case, he's tracking two characters at the same time. So his first, or his parental generation, he took two true breeding parents that were different in two characters. And when he crossed these two true breeding for two different character parents, when he crossed them together, he created the F1 generation, which are now called dihybrids because they're hybrids for two different characters. And then he took the F1 generation dihybrids, crossed them to themselves. This is called a dihybrid cross. And in this case, the F2 generation outcome allowed him to determine whether the two characters that he was following were traveling together as a package deal to the next generation, or if the genes for these two characters traveled independently of each other to the next generation. So that's a lot of words. Let's take a look at an example. Now we're going to spend some time with this slide. Make sure you understand it and the different outcomes and how this brings up Mendel's laws of inheritance. If not, please by all means ask questions. Okay, so we've got the same layout as his single character crosses. We've got the P generation first. They're crossed to each other to give the F1 generation, and the F generation crossed to themselves to give the F2 generation in this big box. Now you'll notice this big box has two outcomes. The one on the left is the hypothesis that, well, maybe the two characters that Mendel was following, they traveled together to the next generation from the F1 to the F2. The other side, on the right-hand side, this is the outcome that you would get if the genes for those two characters sorted themselves independently, traveled separately. And so let's go through step by step on how he set up these crosses and what were the results that he got. So in this case, he's looking at two different characters. He's looking at seed color. Peas can be either yellow or green. The yellow P's are going to be the dominants, the green are going to be recessive. The other character that he is following is seed shape. So P's can either be round, this is the dominant character trait, or they can be wrinkly. Wrinkly, these are going to be the recessives. So he's taking his parental generation P's here. So he has a homozygous dominant for both characters. So this P is yellow and round, has two homozygous or two dominant yellow alleles, and he has two dominant round alleles. The other parent is recessive, homozygous recessive for both characters. So it's a green and wrinkly P. It's got two recessive green alleles and two recessive wrinkly alleles. So when these two parental generation P's make their gametes, remember each gamete only gets one copy of every gene. So one copy of a color gene and one copy of a shape gene. The yellow and round P has only dominant alleles to give, so all gametes will have a dominant yellow and a dominant round allele. The green and wrinkly P has only recessive alleles to give, right? So in this case, every single one of the gametes will receive a recessive green and a recessive wrinkly allele. When you cross these P generation organisms to each other, you end up with your F1 generation dihybrids. They are heterozygotes for both characters. So one dominant and one recessive allele for the color gene, and one dominant and recessive allele for the shape gene. So far, that shouldn't be too difficult. The next step was to take these dihybrids and do a dihybrid cross. Essentially cross them to themselves to get the F2 generation offspring. So here was Mendel's question. He wanted to know, well, the color gene and the shape gene, do they travel as a package deal, or do they travel separately when these genes are passed on to the next generation? If they travel together, they're stuck together, that means that the same way that these genes were inherited from the P generation would be the same way that the F1 generation would have to pass them on to the F2s. Sorry, clicked too soon. Okay, so in this case, you would have gametes for the F1 generation dihybrids. 50% of them would have the two dominant alleles and 50% would have the two recessive alleles, because this is the same way that those genes were inherited from the P generation. So you would have a 4-square Punnett square for both. The sperm would have two options, and the egg would also have two options for gametes, two dominant alleles or the two recessive alleles. And so in this case, we would get the same result as what we saw in our monohybrid crosses with the 3 dominant alleles, or dominant traits, let's say, for every 1 recessive trait. The other option that Mendel was testing was, well, maybe the 2 dominant alleles don't have to travel together and the 2 recessive alleles don't have to travel together. Maybe you can have a mix. So if the alleles can be mixed, that means you can match up any color allele with any shape allele and vice versa. So for every sperm, there would be 4 different possible options of gene combinations. You could match a dominant shape allele to a dominant color allele. You can see both dominants there. You could match a dominant color allele with a recessive shape allele. Or you could match a recessive color allele with a dominant shape allele, or you can put 2 recessives together. And this would be the same option for the sperm, the same options for the eggs. And so having 4 different possible gamete combinations, now you need a larger Punnett square to figure out, well, what are all the possible offspring that can result from any number of fertilization events. And so this is a 16 square Punnett square, same idea. You would go egg to sperm crosses all the way through to get your results. Just to give you a little bit of sort of technique information, when you're doing a larger Punnett square like this and you're looking at 2 different characters, you always want to keep the 2 alleles for one character together. So the Y's are always together, the R's are always together. And usually we write the dominant allele first and then the recessive. So when you have the heterozygotes, doesn't matter which parent the dominant or the recessive allele came from, you always write the dominant first followed by the recessive. All right, so in this case, with a 16 square Punnett square, where the two genes for color and for seed shape don't have to travel together, the phenotypic ratio comes out to be a little bit different for the potential offspring that could result. Here you would have a 9 to 3 to 3 to 1 phenotypic ratio. And what's interesting is you'd have some of these hybrids in the middle, right? You started out with parents that were round and yellow and green and wrinkly. And now having this independent assortment of these different genes, you could end up with a green round and a yellow wrinkly pea, as opposed to what you started with at the very beginning of these crosses. And so when Mendel did this test, and he did this P to F1 generation to F2 generation cross, he got results that looked something like this, where he saw 315 yellow and round, 108 round and green, 101 yellow and wrinkly, and 32 green and wrinkly. And he got that approximate 9 to 3 to 3 to 1 phenotypic ratio, supporting the idea that the genes for seed color and seed shape don't have to travel together when they are passed on to the next generation. So using this dihybrid cross, Mendel was able to develop his second law of inheritance, which is the law of independent assortment. The law of independent assortment states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. So the pair of color alleles are going to separate independently of what the pair of shape alleles do during gamete formation for those dihybrid peas that we looked at in the previous example. Now there's a catch to this law of independent assortment. It only applies under certain conditions. So law of independent assortment is only going to apply to genes that are on different non-homologous chromosomes, or those that are really far apart on the same chromosome. Genes, on the other hand, that are located near each other on the same chromosome, they actually tend to be inherited together. They are linked, and they will be passed on to the next generation kind of as a package deal. This last point here kind of makes sense if you think about it, right? If you're in the same car, if you're on the same vehicle, you're probably all going to end up at the same destination, right? If the genes are close to each other on the same chromosome, when the chromosomes are sorted and they move during gamete formation, during fertilization, if you're on the same vehicle as genes, you're probably going to end up in the same eventual offspring or cell. Now, going back to the previous point, if you're on different chromosomes, right, different non-homologous chromosomes are going to do their own thing, right? You're in different cars, you can end up at different destinations. That makes a lot of sense, right? Those genes on different non-homologous chromosomes are going to be sorted independently of each other. Where the catch is, where it's a little bit tricky, and I encourage you to think about this before we come back to it in a later chapter. What about those genes that are on the same chromosome, but they're really far apart? Is there any way that they can be unlinked from each other during gamete formation? So think about it. We'll come back to it.